Emerging and re-emerging pathogens are global challenges that very likely will happen again in the future. Considering the severe acute respiratory syndrome coronavirus, SARS-CoV, outbreak in 2002, and the Middle East respiratory syndrome coronavirus, MERS coronavirus, outbreak in 2012, the present outbreak of a coronavirus-associated acute respiratory disease called coronavirus disease 19, or COVID-19, is the third documented spillover of an animal coronavirus to humans in only 2 decades that has resulted in a major epidemic. Using the isolated SARS coronavirus 2, researchers have identified a critical host susceptibility factor. When culture cells overexpress the transmembrane protein angiotensin-converting enzyme 2 from humans, those cells become hypervulnerable to infection, showing that this protein works as SARS coronavirus 2 receptors. These findings resemble back to the earlier SARS coronavirus, which also utilizes both human and animal ACE2 proteins as receptors. SARS coronavirus, SARS-CoV, uses angiotensin-converting enzyme 2 as a receptor and primarily infects ciliated bronchial epithelial cells and type 2 pneumocytes. Type 2 pneumocytes, shown in this diagram, are cuboidal cells that line the lung alveoli and are responsible for producing pulmonary surfactant, which decreases the surface tension by decreasing the density of water molecules at the air-water interface, which substantially ease the expansion of the lungs during ventilation. The most threatening bat-derived coronavirus are those with distinctively human-tropic S proteins. Once inside human lungs, S proteins interact with host susceptibility factors, including the ACE2 receptors, which trigger virus cell membrane fusion and infection. Therefore, the spike protein of coronavirus facilitates viral entry into target cells. The receptor binding domains on the SARS coronavirus 2 S proteins bind with a high affinity to human ACE2 receptors, which likely explains the ease of transmissibility of this virus. After initial reports of SARS-like virus emerging in Yuan, it appears that this new virus is actually less pathogenic than MERS-CoV and SARS-CoV, MERS coronavirus and SARS coronavirus. Age and coexisting conditions such as diabetes or heart disease are independent predictors of adverse outcomes in SARS coronavirus and MERS coronavirus. Emerging viruses that may go undetected because of the lack of severe disease in healthy people can pose significant risks to vulnerable populations with underlying medical conditions. In a retrospective study with data regarding 1,099 patients with laboratory-confirmed coronavirus COVID-19 disease from 552 hospitals in 30 provinces, autonomous regions, and municipalities in mainland China through January 29, 2020, researchers found the median age of the patients was 47 years. From the 1,099 patients, 5% were admitted to an intensive care unit, 2.3% underwent invasive mechanical ventilation, and 1.4% died. The most common symptoms were fever, 43.8% on admission and 88.7% during hospitalization, and cough, 67.8%. Diarrhea was uncommon, only 3.8% of the patients. On admission, the degree of severity of COVID-19 disease was categorized as non-severe in 926 patients and severe in 173 patients. Patients with severe disease were older than those with non-severe disease by a median of 7 years. Moreover, the presence of any coexisting illness was more common among patients with severe disease than among those without. Therefore, age and coexisting conditions such as hypertension are independent predictors of adverse outcome. Despite the number of deaths associated with COVID-19, SARS coronavirus 2 appears to have a lower case fatality rate than either its earlier family members, SARS coronavirus or MERS coronavirus. The case fatality rate in a real-world scenario may very well be much lower than 1.4% because patients who were mildly ill or asymptomatic did not seek medical attention and therefore were not included in any study. The case fatality rate seems to be age-dependent, with a higher percentage in the elderly, which is basically true for any disease. The number of individuals with undetected mild case could of course be much higher than the official case number, which would lead to a lower fatality rate per infection.